Hello everyone. So in this session, I'll be going over high yield topics of the NEET PG, upcoming NEET PG 2022, right? And if you want me to present more topics other than these I have provided to you, right? And other than those which I have given you the previous year also, right? So you can just make sure you drop me an email or just send me a message on my Telegram group, or you can add yourself in a Telegram group also. We do lots of discussion there, right? So I went over lots of like recall questions for the NEET PG, and I figured out the urea cycle. Urea cycle is must know topic. There are so many things that are involved. So these are the like few questions that were there. Hyperammonemia, right? Hyperammonemia is there is elevated levels of the ammonium ions. It inhibits the TCA cycle, right? So there is this interrelation between the urea cycle and the TCA cycle. They are interrelated with each other. How does the hyperammonemia means the ammonium ions are not converted into urea by the urea cycle, that is leading to the increased ammonium ions. They inhibit the TCA cycle. What do we know about this? Right, ammonium ions actually, if they are in abundance, they combine with alpha ketoglutarate, right, and they forms glutamate. There is this enzyme glutamate synthase, right, that synthesizes or the glutamate, right. Again glutamate can combine with one more ammonium ion, right, and it can form glutamine, right. There is another enzyme that is present which helps in the synthesis. So ammonium ions, increased ammonium ions are like body try to compensate it by forming a glutamine. So what is the big catch here? If there is lots of ammonium ions, they will be used up by the alpha ketoglutarate, resulting in the depleted levels of the alpha ketoglutarate, which is required for the TCA cycle, right? So alpha ketoglutarate will not be there. That is going to halt the TCA cycle, right? So answer to this is choice B. Hyperammonemia decreases alpha ketoglutarate and thereby inhibiting the or hampering the TCA cycle, right? So there was another question related to the urea cycle. In urea cycle disorder, which of the following substance can be used to reduce the ammonium levels, or you can say the ammonium ions level? So in the previous question, what we have saw, ammonium ions combined with alpha ketoglutarate to form the glutamate, right? So there is a high level of the glutamate that is already present, right? And then they are combining with one more ammonium ions, and finally what we are having glutamine in the system, right? So whatever drug or therapy we have to provide to reduce the urea level, that compound should combine with the glutamine. So that is phenylbutyrate. Okay, so glutamine combines with phenylbutyrate, butyrate, right, and it forms phenylacetyl glutamine, and this compound is soluble and then it can be excreted via kidneys, right. So phenylbutyrate is given for treating the to reduce the level of ammonia or to treat the hyperammonemia, right. Now there was one more question from the choices given. Which pair is the source of nitrogen in the urea cycle? Do we know the structure of the urea? Urea is carbonyl carbon, and on both sides we have the NH2 group present, right. This is typical of urea, right. One nitrogen is coming definitely from the ammonium ions because that what need to be excreted, and this carbon coming from carbon dioxide. There is this another third nitrogen that comes from the aspartate, right. So there are so many things we should be knowing about the urea cycle. So first of all, I would like to give you a brief about the urea cycle. What we have in urea cycle? Urea cycle, you have the carbon dioxide, you have the ammonium ions, right, and they together form carbamoyl phosphate, right? They together form the carbamyl phosphate. And what is the enzyme that is involved here? CPS1, right? Carbamyl phosphate synthase 1. And this is what? This is a rate limiting enzyme, and this is the rate limiting step of the urea cycle, right? Now we know there is another enzyme CPS2, right? That also forms carbamyl phosphate, right? So what is this difference between these two enzymes? Here there is the carbon dioxide, and what else combines here is there is some other thing that combines, and this cycle leads to what? The pyrimidine synthesis. Urea cycle first step is taking place in mitochondria. We have 5 steps in the urea cycle. 5 steps. First 2 steps, step number 1, and there is a step afterward that takes place in the mitochondria. So we can say CPS1 is what? CPS1 is a mitochondrial enzyme, whereas CPS2 is what? CPS2 is a cytosolic enzyme, right? Or as I always say in the class also, for the examination point of view, regulation is very important. So here the rate limiting enzyme in the urea cycle is a CPS1, right? But there is a similar isoenzyme as CPS2 also, right? That is for pyrimidine synthesis. And there the regulation is how nature has made the compartments different. So there is a compartmentalized regulation of this, right? Urea cycle first step is in the mitochondria, whereas pyrimidine this step is in the cytosol. That is how things are not getting mixed up. So this is CPS1, this is CPS2. Just know this, right? Now coming back to our urea cycles, carbamyl phosphate that combines with something. There is something that is combining, and that is going to form what? Going to form C carbon dioxide, C carbamyl phosphate, and another C citrulline. Another C is citrulline. All C's are there: carbon dioxide, carbamyl phosphate, then citrulline, right? And what is combining here? This ornithine. There is a substrate ornithine that combines with the carbamyl phosphate to gives you the citrulline. Since ornithine is combining, the name of the enzyme is ornithine transcarbamylase. The enzyme is ornithine transcarbamylase, and again it is a mitochondrial enzyme. Deficiency of either of these two are going to be called as a urea cycle defect, and we will discuss that, right? Now citrulline, this OTCase is what? It is a mitochondrial enzyme, but this citrulline comes out from the mitochondria into the cytoplasm, right? And what is there needed? There is a transporter that is needed, right? Or there is an enzyme permease, right? Permease that is there, available there, which transports, or we can say that allows citrulline to be present in the cytoplasm so that further urea cycle can be carried out, right? So if there is a defect with the permease, that is going to elevate the levels of the citrulline, right, in the mitochondria, giving rise to citrullinemia, okay? That is going to give rise to the citrullinemia, right? Now this citrulline combines with another nitrogen source. What was the another nitrogen source? Another important question asked in the exam: aspartate, right? Second nitrogen of the urea is coming from the aspartate, forming argininosuccinate, right? And since we are synthesizing argininosuccinate, so enzyme name is argininosuccinate synthase. Argininosuccinate synthase, right? And this is a cytoplasmic enzyme, right? And again it is going to lyse, it is going to break down into 2 halves, right? So the enzyme name is enzyme argininosuccinate lyase. So we are breaking the argininosuccinate into 2 parts, and then what will we get? We usually get arginine. One thing is arginine for sure, right? That is be helping us to carry out the synthesis of the urea. Plus another thing is a fumarate. Where do we see fumarate? Fumarate we see in relation to this TCA cycle. Fumarate can be converted into malate Then malate can be converted into what? Oxaloacetate, am I right? And this all thing, this part looks very similar to the TCA cycle, right? So don't get confused here. Why? Where we are? We are in the cytoplasm, right? And TCA cycle happens in the mitochondria. So this is a similar kind of a process that takes place in the TCA cycle, but this is cytoplasmic, not the mitochondrial, right? Be careful with that in exam also. They may trick you because the exam is getting trickier every day. So from arginase, we synthesize arginine. There is enzyme arginase, right? Arginase that acts upon it to synthesize what? To synthesize urea. What else you get? We get, what do we get? Come on, what do we get? What we have used up there in the pink? Ornithine. So here we get our ornithine back. Here we get our ornithine back, which can go up there again, right? This is the urea cycle, and there are 5 steps: 2 mitochondrial, 3 cytoplasmic. There is one permease that is involved, right? And then there is another sideways cycle, fumarate, that is similar looking steps, that follows a similar looking step of the TCA cycle, but TCA cycle is mitochondrial, whereas all these things are happening in the cytoplasm. So that is contributing to the cytoplasmic levels of the oxaloacetate. In other words, you can say so, right? We are not going to discuss beyond this here, otherwise you will get confused. So these are the enzymes. Regulation is important for the examination point of view, right? So what is the regulatory enzyme? CPS1 is the regulatory enzyme, right? What is the regulatory enzyme? CPS1, very first enzyme. Who controls CPS1? Who controls CPS1? This, who controls it? There is NAG. There is this compound N-acetylglutamine, NAG, right, is a positive regulator of CPS1. Who synthesizes it? N-acetylglutamine synthase enzyme. NAG's enzyme helps in the synthesis of the NAG. So this is one regulation. If there is a defect in this enzyme, that is going to halt the synthesis of the NAG, so CPS1 will not work. So this is one way to regulate it, right? NAGS deficiency. So in the urea cycle, whatever deficiencies you have in the urea cycle, all these are what? Hereditary, right? Or in other words, you can say if you have hyperammonemia, right? Hyperammonemia can be during the lifetime, that is not inherited, right? That is maybe because of the liver issues, there is a damage to the liver, right? That is contributing towards the elevated level of the ammonium ions. But if there is a defect in the urea cycle, all these will lead to hyperammonemium conditions, right? And that is genetic. So CPS1 defect, if we talk about defect, CPS1 defect, enzyme defect is rare. It is rare. CPS1 enzyme defect is rare and it is positively regulated by N-acetylglutamate, right? And N-acetylglutamate is synthesized by N-acetylglutamate synthase, right? So if let's say a person is suffering from urea cycle defect, and if we can provide N-acetylglutamate in the diet and the urea cycle disorder gets corrected, so we know that the NAG was not forming, there is a defective NAG synthase. That is another important question, right? This is another important question. The CPS1 enzyme, CPS1 is okay, but still urea cycle is defective and the person can respond well to the what we are giving, well to the NAG in the diet. They respond well to the NAG in diet, that is telling there is not CPS1 deficiency, but there is NAG synthase deficiency that is happening. Okay, so CPS1 deficiency is number one. I told you first enzyme, that is rare. This is a rate limiting enzyme, right? And the another enzyme was OTCase. This is not a rate limiting enzyme, but it is very important enzyme, right? and this inheritance is X-linked inheritance, and there will be elevated level of glutamine in these patients. Elevated level of glutamine in the blood, in the CSF, and also in the urine. So there is another important thing that is related to the OTCase enzyme. If we come back to the urea cycle, what do we see here is? What do we see here? If let me erase everything here, up here. See here. If this enzyme is defective, so this will not take place. This is not going to take place. But what you will be having here? You will be having the elevated levels of the carbamyl phosphate. Right, we will be having the elevated levels of the carbamyl phosphate, then that will keep on accumulating in the mitochondria, and then it is gonna spill out in the cytoplasm. Where we have seen the carbamyl phosphate, it will come out in the cytoplasm and it will go to the synthesis of what? It will contribute towards the synthesis of what? The pyrimidine synthesis, or you can say the purine pyrimidine synthesis. Now, if you take a look at it, what you will see is this carbamyl phosphate is going to have, is going to add on to the orotic acid levels, because after carbamyl phosphate is formed, there is a formation of orotic acid, right? This level will be elevated and the person will be suffering from, the key word I wanted to highlight here is, the key thing is the person will be suffering from orotic acid urea, which was not the case when the person was having defective CPS1 deficiency, right? So if we have to compare CPS1 deficiency with the OTCase deficiency, right? Let's say if I compare these two, CPS1 and OTCase deficiency, right? In both the cases, there will be elevated levels of ammonium ions. There will be elevated levels of ammonium ions. Hyperammonemia, there will be decreased burn level. There will be increased glutamine level. But in CPS1, there is no orotic acid urea, which will be present here, right? So that's how we can distinguish between CPS1 and the OTCase deficiency, right? So we know now how to distinguish between CPS1 deficiency or an NAGS deficiency. Now we know how to distinguish between CPS1 and the OTCase deficiency. What was the another important thing here in the urea cycle? This enzyme. Which enzyme? The permease enzyme, right? If this enzyme is defective, right, it is not allowing citrulline to come outside. So what it will lead to, right? What it will lead to? That will lead to three conditions. HHH, right? Hyperornithinemia, hyperammonemia, and homocitrullinemia, right? All three things will be happening. HHH, that is because of this permease enzyme, which is also known as ornithine permease, right? Why? Because this will be accumulated, right? And ornithine that we are getting back, regenerating back, right? That is also accumulating here. And also there is hyperammonemia, because that is going to feedback inhibit this enzyme and the ammonium levels will be elevated as well, right? So this is the fourth deficiency, very important for the urea cycle. Now what happens if there is arginine succinate synthase enzyme deficiency? If this is deficient, this is not working, there will be again citrullinemia. There will be elevated levels of the citrulline where in the cytoplasm, that can be detected, okay? And if there is this lyase enzyme deficiency, there will be elevated levels of the argininosuccinate, right? And if there is deficiency in the arginase enzyme, there will be elevated levels of the arginine, which can be detected, okay? So these are the various urea cycle disorder defect. Most important ones are the upper one, the CPS1, OTCase1, right, and the NAGS1. This is these are very important for the examination point of view, right? this is related to your dermatology. That is why I have brought this up again. The succinate synthase, arginosuccinate synthase deficiency was leading to the increased level of the citrulline, right? And it is what? It is the second most common defect of the urea cycle. Why I am bringing it up? Because in the previous questions, recall questions, I figured out there was a question revolving around trichorhexis nodosa, like the tufted hair you see here, tufted hair. That is because of this enzyme deficiency, right? It is a common hair problem. It is a common hair problem in which the thickened or the weak points along the hair shaft cause the hair to break off, right? The hairs, the break off very easily. This was one of the recall question, and that is related to the arginosuccinate synthase enzyme. That's why I have brought this up, okay? Now there was another important topic from the various questions about the collagen, right? How we are synthesizing the collagen, what are the important you can say coenzyme cofactors that are involved, right? And what kind of deficiency diseases we can have because of collagen is not formed properly, right? So collagen is one of the very important constituent of your extracellular matrix, right? So most abundant amino acid that is found in the collagen. In the collagen, when we talk about, majorly we talk about 3 amino acid: glycine, proline, lysine. Glycine, proline, lysine. Glycine is fixed all the time. Proline, lysine. If you can recall from the class, we always write glyxy, glyx, glyxy, glyxy. Am I right? So every third amino acid is what? Glycine, that is fixed. So the most abundant amino acid that is found in the collagen is glycine, right? One thing. Now we have to know how the collagen is being synthesized. What are the various steps? So synthesized, the various steps that are involved, various coenzymes that are involved, and which step is taking place where, right? And if the cofactor is or coenzyme is not present, what kind of deficiency diseases we see, right? That becomes a part of a big disease, right? So this is a collagen, typical collagen picture, right, from your Harper. So you have the collagen fibril that is composed of so many fibrils, small like thin fibrils. They are bundled together. You can imagine a process how we synthesize a rope, right? So they are bundled together. The smallest we can think of the triple helix, right? So you have one helix, that is a right hand helix, then you take another helix, then you take third helix, 3 right hand helix, and then they are left handed bundled together, and these many of these triple helices are arranged in a particular fashion, so it forms a very strong collagen fibril, right? And this is the amino acid sequence: glyxy, 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 right? And what kind of a chain is there in the alpha helix? Remember this word also, there is alpha chain that is there. So when we talk about the synthesis of the collagen, right, I'm going to guide you one by one just to save time so we can cover more topics, okay? That's why I have taken this picture, right? So it is divided into 2 phases. Major collagen synthesis is divided into 2 phases, right? This is mitochondria. So things are taking place in the mitochondria first of all, right? In the mitochondria, what do we get? We get a raw collagen, right? Raw collagen, that is a protein that is after like the transcription and the translation, you get what? Pre-pro alpha chains. Like pre-pro insulin we have done, this is pre-pro alpha chains. Alpha chains, right? Up there, what we have figured it out? The collagen is composed of alpha chains. So pre-pro alpha chain we get, and when it exits from the mitochondria, what kind of form we have? We have the pro-collagen. Right, the pro-collagen comes out, and there are various steps that is happening inside the mitochondria, right? That is this yellowish color highlighted thing. What is going on inside? Major 2 steps you should be knowing, right? Actually 3 steps you should be knowing. First of all, signal sequence is being cleaved. Signal sequence is like more or less your extra corners are being trimmed off, okay? signal sequence is being cleaved, then what happens? Then there is a hydroxylation that takes place. Hydroxylation of selected lysine and the prolines, right? Selected lysine and the prolines, and the enzyme for the lysine is, you have to know their name, lysyl hydroxylase. Lysyl hydroxylase, and for the proline it is prolyl hydroxylase. What is done? Hydroxylation is done, and we know if there is this group, right, that is involved in the hydrogen bond formation. So if there are more hydroxyl groups, so they can make the more hydrogen bonds with the nearby residues, that is going to make it more strong. That is the purpose of the hydroxylation. But for exam, what we need to know, the name of the enzyme, and the enzyme is vitamin C dependent. Enzyme is vitamin C dependent. Why that is important? Because what we have seen in the scurvy, bleeding gums, right? Scurvy is bleeding gums. Vitamin C deficiency, am I right? Vitamin C deficiency causes the bleeding gums because here the collagen formation is now don't have this enough of vitamin C. So the fibrils are not formed, they are formed like in the loose form, and the blood comes out, right? So vitamin C deficiency is impairing the collagen synthesis. Where? Because the vitamin C is necessary for the hydroxylase enzymes. Which hydroxylase enzyme? Prolyl hydroxylase and the lysyl hydroxylase. Very, very important point for the exam, right? And after that, selected residues are lysine, not the proline, just the lysine, mind it, not the proline, but the lysines are glycosylated. Glycosylated, we are adding a carbohydrate group, maybe a glucose or a galactose group we are adding, right? And then a pro-collagen forms. So we had pre-pro alpha chain on the top. Now after hydroxylation, glycosylation and signal peptide cleavage, what we get? Pro-collagen we get. So 3 steps happening in the mitochondria. Now this pro-collagen comes out from the mitochondria, and then again few thing happens, modification happens, right? First is terminal propeptides are cleaved. Terminal means these propeptides are being cleaved, because we have to keep the middle segment that is needed for us, right? And then they form the collagen fibers. There is one more step that is happening here. What is that step? There is this enzyme lysyl oxidase. Lysyl oxidase, that is a copper requiring enzyme. So in case of the Menkes disorder, when there is copper deficiency, this lysyl oxidase is hampered, again impairing the collagen synthesis, right? So this lysyl oxidase is also involved. That is another important topic for the collagen biosynthesis formation. Okay, so this is, I have typed it for you guys, right, not written, so that sometimes you are unable to understand my handwriting. So it is divided into 2 halves, the entire collagen biosynthesis, intracellular, extracellular, right? In intracellular, cleavage of the signal peptide, that was one of the recall questions. Hydroxylation of the proline and the lysyl residues, right? That was interrelated with the scurvy vitamin topic, right? They have asked this question also, right? And what do we form? The triple helix. Another asked question was the what? This, related to this, where the amino or the carboxy, because terminal peptides are cleaved. Where, what is terminal? Terminal is your C terminal and N terminal, that is protein, right? So where is that is being removed? So don't get confused between the signal peptide and now here this terminal propeptide. Signal peptide is in the beginning, when we have in the beginning, very first step. Now this is the terminal peptides we are trimming off, right? And now another enzyme, another question, this was another question, this lysyl, right? This lysyl oxidase is there, and what this lysyl oxidase, if question paper is very, very difficult, only then they are going to ask you this, right? The oxidative deamination of the amino group, right? In the lysine, you have the epsilon amino group, if you can recall from the structures of the amino acid. So epsilon amino group is being removed, right? They may ask you, but only in the case if the question paper is said to be very difficult, okay? Now this is the question, one of the recall question, right? 14-year-old boy is undergoing evaluation for the hearing loss, right? And the genetic analysis is showing there is a mutation of type 1 collagen alpha 2 chains, right? Now question is asking you which of the following processes occur outside of the osteoblast where the collagen synthesis is taking place. Now this step is outside the osteoblast. What is being done? C-terminal propeptide is being removed, right? C-terminal propeptide is being removed. That is what is happening. Now, taking further, like the collagen synthesis, I'm going to take it further. Collagen is of various subtypes, right? Call 1A. Just I'm going to zoom in. Collagen 1A1, collagen 1A2, collagen 2A1, 3A1, so on and so forth. There are so many things, but we have to just do the selective and the smart study at this point in time. So what we should be knowing is, first of all, col A1A1 and col 1A2, right? These two. Defect in any of these two genes will lead to osteogenesis imperfecta type 1, right? This is one point. Another thing is collagen 7A1 gene, col 7A1 gene. This is another one of the recall questions that is related to the epidermolysis bullosa, okay? And now, if you will see, there is this ED, Ehlers-Danlos syndrome, not the erectile dysfunction, but the Ehlers-Danlos syndrome, that might happen because of the various type of the defect in different kind of the collagen 1 genes, right? It may happen because of the 1, it may happen because of the 3, it may happen because of defective in the 5 or 3A, or also in case of the defective lysyl hydroxylase enzyme, right? So all these leads to the various kinds of Ehlers-Danlos syndrome. Question: do we need to know the subtypes of Ehlers-Danlos syndrome? Answer from me is really, I don't think so, because I haven't seen this much level of like higher level of the question in the question paper so far. Okay, so just know Ehlers-Danlos syndrome is of various type, and that might be because of the col A1 or 3 or the 5 or the lysyl hydroxylase enzyme deficiency. So I have summarized this chart into a easier last minute point for you, right? Because of the collagen synthesis, there can be two kind of a disorder, right? One which is not hereditary, and another's are the hereditary disorders which have been asked previously in the exam. Which is not genetic? The scurvy, vitamin C deficiency, right? Vitamin D deficiency is hydroxylation is impaired. That is leading to what? The pitaceae, ecmosis, loose teeth and the bleeding gums, right? Loose teeth and the bleeding gums, these are the hallmark for the scurvy. Poor wound healing, poor bone development, right? Now osteogenesis imperfecta, right? There is mutation in the collagen genes. Which one? Col 1A1, right? 1A2 that we have just studied, right? Leading to the skeletal deformities, blue sclera, as well as the fractures, which you have studied in detail in the osteo class also, right? Now the Ehlers-Danlos syndrome can be because of the mutations in the various collagen genes, type 1A, type 3A, type 5A, right? And also the proline and the lysyl hydroxylase enzyme, right? This was one of the recall questions. What are the hallmarks? Hyperextensible fragile skin, right? Hypermobile joints, dislocation, right, and varicose veins. Third is the Menkes disease. There is deficiency of what? Copper, and that copper is required by the lysyl oxidase enzyme. Lysyl oxidase enzyme, and that leads to the depigmented steely hair, right? This is the biggest hallmark. Steely hair, right? Question: Menkes disease is associated with which type enzyme deficiency? Answer to this is lysyl oxidase, not the hydroxylase. Hydroxylase is related to the ED, Ehlers-Danlos syndrome, right? I want to show you the pictures also that is related to these kind of the disorders, that is related to the collagen synthesis, right. The first one is the Ehlers-Danlos syndrome, right. Ehlers-Danlos syndrome is also referred to as cutis hyperelastica. Hyper means elastica, more than elastic it is, right. Cutaneous, like cutis is word coming from the cutaneous, that is skin. Hyper, too much of the elastica is elasticity. So cutis hyperelasticity, and which genes are involved? Type 1, 3 and 5s are involved, right, and that is leading to the hyperextensibility of the skin, abnormal tissue fragility and increasing joint mobility, and they may give you this kind of a picture or somewhat like related pictures for the Ehlers-Danlos syndrome, right. There is another very important syndrome, that is Alport syndrome, also known as hereditary nephritis, right. That is because of type 4 collagen synthesis is impaired, right, and the presenting sign is hematuria accompanied by the ocular lesions, right. This is Alport syndrome. Epidermolysis bullosa, one of the recall questions previously they have asked, that is epidermolysis is 7A1, right, 7A1 gene, and there is another variant of this, but that is epidermolysis bullosa simplex, but that is related to the mutation in the keratin. Epidermolysis bullosa is the collagen, whereas epidermolysis bullosa simplex is the keratin. So don't make a mistake in the exam, okay. Now the Menkes disorder, right, the clinical representation, what you will see, the baby, 7-month-old infant, what it had? Abnormally kinky and hypopigmented hair, right, and what is? Low levels of serum ceruloplasmin. Ceruloplasmin is related to your copper levels, right. Only 10% of the normal serum copper levels are there. It is more common in males. Gene involved is, why I am giving you this much, because they may ask you the any way they can ask you what is the gene involved? ATP7A. Like ATP, you can remember the adenosine triphosphate wala, right. ATP7A gene is involved, right, because it encodes what? ATP dependent copper efflux protein, right. To let the protein out, that ATP is required. So ATP dependent copper efflux protein, there is this protein, there is this efflux protein which is effluxing the copper out, but that protein is ATP dependent, that is why the name is ATP7A, and there is no cure and life expectancy is this much. Now here don't get confused with Wilson's disease. Many a time many students they do. Wilson's disease is excess level of the copper ions, right. Menkes is the lower levels of the copper ions, like this is the steely pigmented kinky hairs, right, and that is defective cross-linking of the collagen and elastin by copper-dependent enzyme lysyl oxidase, that is leading to all these defect of the Menkes disorder. Now another very high yield topic, as you can see from this picture, I have written Hartnupp's pellagra carcinoid syndrome, right. What do we know about the pellagra? We know the Cazals necklace. Almost every other exam they are asking you, they are giving you something with this Cazals necklace, right, and the patients they are on the maize or sorghum eaters, right, sorghum eaters, that is leading to the deficiency of the niacin. How do we synthesize the niacin? Niacin is one of your atypical enzyme, remember, tryptophan. 60 milligram of tryptophan gives you 1 milligram of the niacin, right, and tryptophan is your what? That is why tryptophan is your essential amino acid, EAA, essential amino acid. If tryptophan is impaired, you don't have it, there will be decreased level of the niacin, that is going to give rise to the pellagra. Pellagra is 3Ds or the 4Ds, dermatitis, dementia, and what else is there? Dermatitis, dementia and diarrhea, right, and sometimes they write death also, right. This was one thing Cazals necklace, they give you always and always they give you one question. Somehow they give you. But there are two other diseases. You have to pick the correct choice. There is a difference between the exact right and the very right, right? So you have to pick the exact right choice. So there is the Hartner's disease and the carcinoid syndrome, in which the patient shows the pellagra-like symptoms. But the difference, the cause of the disease is different, right? In the purely pellagra, niacin deficiency, right? The problem is to synthesize the niacin from the tryptophan. Maybe one of the enzyme is deficient, right? In Hartner's disorder, there is failure to absorb the tryptophan, right? Because what is defective? No, the transporter is defective. So tryptophan in the diet is failed. It cannot be absorbed by the body. Hence the niacin cannot be synthesized, leading to the pellagra-like condition. So know the difference of the what is causing the Hartner's disease. What is causing the carcinoid syndrome, right? Carcinoid syndrome, either TRIP can follow this pathway to form the niacin, right? Or it can form to make serotonin. Serotonin, remember tryptophan is the name. TOTO, I always say TOTO, it forms serotonin, it forms melatonin also, tonin. Remember this way, serotonin, melatonin, right? So if this entire tryptophan is routed towards the formation of the serotonin, none is available for the synthesis of the niacin. That is your carcinoid syndrome, okay? So this is another very high yield topic. Pellagra, they always ask you one question in either way, right? So I have given you the entire bag for that. Just know this. Now few important topics, questions from the purines and the pyrimidines metabolism. Purine and pyrimidine metabolism, right? What we know about the purine? First of all, know the basic structure. Small name, big structure, as I always say. Small name, big structure, right? Pyrimidine is bigger name, so small structure. So this is the one way you can figure out the difference, whether they have given you the structure of the purine or the pyrimidine, right? That is gonna save you time. Purine is pure as gold. So adenine, guanine. Pyrimidine is cut the pie, cytosol, uracil, thymine, okay? So these are the basic things of this, purine and the pyrimidine. Purine is synthesis. Purine we can synthesize either by de novo or salvage pathway. De novo is from the scratch. Salvage is we have recycled few components and we can reuse in the synthesis of the purine, right? In the pyrimidine, what we have seen in the urea cycle, also what we have seen, carbon dioxide plus in there you had ammonium ions, but here one of the nitrogen is coming from the glutamine. They ask you this question. Remember in the class, just go and check the notes, which atoms are contributed by what. That is another high yield topic. So carbon dioxide and glutamine, they makes carbamoyl phosphate, and the enzyme here is CPS2, and this is a cytosolic enzyme, right? And this carbamoyl phosphate, over there carbamoyl phosphate was combining with the ornithine to give you citrulline. Here this carbamoyl phosphate is going to join to another amino acid, and that amino acid right here is aspartate, and that forms the orotic acid, right? If there is a deficiency in the CPS2 enzyme, that is going to give rise to like what you will be having. If there is this defective enzyme, then there will be increased glutamine level, right? If there is too much of the carbamoyl phosphate, why carbamoyl 2 phosphate is in excess? Because in the urea cycle OTCase is deficient, it is not working properly, so carbamoyl phosphate formed inside the mitochondria comes outside in the cytoplasm, leading to more increased level of carbamyl phosphate, hence the increased level of the orotic acid giving rise to orotic acid urea, right? And then what do you do in the pyrimidine synthesis? Pyrimidine is CUT cut. First was UMP, and that is not deoxy. That should be converted into UDP and then deoxy UDP, right? Deoxy UDP. From there, deoxy UDP, what is the enzyme? Another important topic, DUDP. Deoxy means what we are doing? We are doing the reduction. So enzyme is a reductase. We are doing the reduction of what? Ribonucleotide. This is a ribonucleotide. So ribonucleotide reductase is the name of enzyme, and this is target from hydroxyurea. This is a target for the hydroxyurea, right? Another important topic, question for the exams. This DUDP then is converted into DUMP. And what do we know? In DNA we don't have U. In DNA we have what? T. So this will be converted into TMP. TMP. The enzyme is what? What we are synthesizing? We are synthesizing thiamidylate. So the enzyme name is thiamidylate synthase. Why I am talking about this enzyme? What we need? We need a methyl group here. That methyl group is donated by N5N10 tetrahydrofolate. Remember, there are two methyl donors, right? This is donated by this, and then what we get is dihydrofolate, which should be regenerated back. Okay, so if there is any deficiency, folate deficiency is impairing thiamidylate synthase enzyme, hence this will not be formed, and then there will be problem in the DNA synthesis. My point is this, will be a problem in the DNA synthesis. So folic acid is related to the DNA synthesis. And who inhibits the enzyme? In case of the cancer, there is this drug 5-fluorouracil, because we don't want the cancerous cells to divide, right? This enzyme mimics the IMUMP, right? And it intercalates so that DNA synthesis is halted in the cancer cells. Another important question for the examination point of view. They have asked you this question in the exam, right? Now, this was the pyrimidine. Now, what about the purine synthesis? Purine synthesis is the de novo synthesis and the salvage synthesis. I'm going to focus on the salvage synthesis because it is involved with 4 or 5 questions that they may ask you, because they are another high yield topics for the exam, right? Just to save the time, I'm using this chart because we have limited time here, right? So here you have adenine and the guanine. This is the salvage path, right? So we are doing the breakdown and then we are reusing some of the components. ATP is converted into AMP, AMP is converted into GMP. Just follow my pointer if you are not able to understand the entire chart, right? This AMP, what is this? This is nucleotide. Nucleotide is composed of what? Nucleotide has a sugar and the base and the phosphate, right? A sugar and the base and the phosphate. If we remove the phosphate, then you are left with the molecule containing sugar and the base. That is not nucleotide, but that is a nucleoside instead. So first what goes? A phosphate molecule is being removed. So what you are left with? A nucleoside. So AMP become a nucleoside. Name of the AMP nucleoside is adenosine. Name of the GMP, that is not containing phosphate anymore. It is a guanosine, right? And from the guanosine we remove the sugar, and what we are left with? We are left with the base, that is guanine. Now this guanine base can be reused for the synthesis of the purine nucleotide, right? Similarly, adenosine first amino group is being removed. A adenosine contains a amino group on the top, if you can recall from the class. You are left with inosine. Then the sugar is being lost and then we are left with hypoxanthine. This hypoxanthine is sort of a base that can be reused for the synthesis of the AMP, adenosine monophosphate. Good. But what we need to know for examination point of view other than this is, other than the pathway, because this is not going, they usually don't ask this. They will ask you what are the enzymes that are being involved. Important enzymes are adenosine deaminase. And another important enzyme is hypoxanthine, HBGRT, right? Hypoxanthine phosphoribosyl transferase enzyme. These are two important enzymes that are involved here. If there is deamination deficiency, right? Deaminase is deficient. That is going to give to SCID, severe combined immunodeficiency disorder. T cells, beta cells, B cells, T cells, beta cells will be hampered. That will lead to SCID. This is another high yield topic and they have asked you this question in the pre-call questions, right? What it is? Adenosine deaminase deficiency leads to the SCID. One question. Another question is HBGRT. HBGRT, right? This enzyme is deficient and that is leading to what? Lesch-Nyhan syndrome. And the patient will be, what we will see? Spastic cerebral palsy. Self-mutilation is the key point. Biting of hands and lips. Hyperuricemia. Gout. Early death, and this is X-linked recessive disorder. Very, very important question for the examination point of view. HGPRT. It is not just they are just biting, they are just making them bleed. So the patient is being held, captured via the chains or the ropes or something, right? So HGPRT, and the biggest keyword you will find it a self-mutilation. Self-mutilation. They may ask you this question about allopurinol also, because this is an integrated question. Allopurinol inhibits what? Xanthine oxidase. Xanthine oxidase. Why this xanthine is coming from? This xanthine is a metabolite of hypoxanthine. When we have to break it down, hypoxanthine is converted into xanthine and finally to the uric acid, right? Or we can say what? Uric acid levels are increased in what? Gout? Increased in gout. So to treat the gout, this allopurinol, which is a prodrug, is given, which inhibits the XAO, XAO, xanthine oxidase, thereby helping to maintain the uric acid levels or decrease the uric acid level in the body to treat the gout. Okay. Now coming back to the Lesch-Nyhan syndrome, I want to give you, there are two conditions that are related to HGPRT deficiency, right? Either the enzyme is partially working, or partially deficient, or it is completely deficient. If this is completely deficient, the disease is Lesch-Nyhan syndrome. If partial, then it is Kelly-Sieg-Miller syndrome. Know this, but this is most important. They usually look for the Lesch-Nyhan syndrome, and these are the various pictures. You may see one of these in the exam also. Biting of the lips, right? The pacifiers are being used. Even the tongue they bite, they bleed, they chew the fingers, right? And because of elevated uric acid, uric acid where it goes? Deposits in the form of the needle shaped crystals, and those crystals are your monosodium kind of a crystals, right? And they deposit in the synovial fluid of the joint. Another question that can come from, where are these for the uric acid testing, what we go for? We go for test the synovial fluid in the joints, right? If there is elevated level of these monosodium crystals which are needle shaped, that is indicating the increased uric acid, right? And the major hallmark of the gout is what we see always a big toe or tophi we see. What is the keyword? Tophi you see. And because of the increased uric acid, patient may experience the kidney stones, right? So this was Lesch-Nyhan syndrome, which was related to the purine and the pyrimidine biosynthesis, right? I want to discuss one more topic before I finish this, that is about glucose 6-phosphatase deficiency. Glucose 6-phosphatase deficiency. First of all, where do we see this enzyme? This enzyme converts glucose 6-phosphate into glucose. Am I right? Right? And this is which topic we are talking about? Glycogen storage disorders, right? And in the question you will see there are elevated levels of the glycogen, or glycogen is being accumulated, right? So the enzyme converts glucose 6-phosphate into glucose. Where do we find this enzyme? This is a part of your gluconeogenesis. Gluconeogenesis, and that means we are talking about the fasting condition. We are talking about the fasting condition, and in the fasting condition what we have? We have hormones. What kind of hormones are elevated? Glucagon. The major hormone, glucagon, is elevated, and maybe cortisol. Right? So glucose 6-phosphate to glucose will be hampered in the glucose 6-phosphate deficiency, and this enzyme is majorly found in actually endoplasmic reticulum. In the endoplasmic reticulum. How does it work? This is the endoplasmic reticulum. You have glucose 6-phosphate that will enter via a glucose 6-phosphate transporter inside. So glucose 6-phosphate will come inside, and then there is this glucose 6-phosphatase anchored to the membrane that is going to convert glucose 6-phosphate into glucose plus inorganic phosphate, okay? And then there is another transporter which will put back the inorganic phosphate into the cytoplasm, and there is this another transporter which will bring out the glucose, and this glucose can be excreted or expelled out in the lumen by the GLUT2 transporter. Right? So in the glucose 6-phosphatase deficiency, we are talking about now two subtypes, majorly two subtypes. One, if this transporter is defective, and another is this transporter is defective, right? So this will give you type 1A and this will give you type 1B. And which is the disease I am talking about? I am talking about the von Gerke's disorder. They ask you questions like in the various way based upon just this glucose 6-phosphatase enzyme, right? So type 1A, type 1B, not 2B but 1B, right? If transporter is defective, right? So this is one piece of information I wanted to give you. So if the glucose 6-phosphatase is deficient, is this patient is going to respond to the glucagon if glucagon is given, right? Since the enzyme is deficient, right, they do not respond to hormones. So if you see something in the question, person is suffering from hypoglycemic condition. Patient is suffering from hypoglycemic condition. Glycogen is being accumulated, right? And they do not respond to the hormones, glucagon therapy, right? Then that we are talking about what? The von Gerke's disorder. One thing to it, okay? Now, if that is one way to look at it, there is another way to look at it also. So it means glucose 6-phosphate cannot form glucose. So there is an elevated level of the glucose 6-phosphate. Where it will go? It will reroute it towards the synthesis of the pyruvate. From glucose 6-phosphate, pyruvate will be formed, and this pyruvate will be converted into what? Acetyl-CoA. Right? Acetyl-CoA is giving you what? It helps in the synthesis of the ketone bodies. So there will be ketone bodies synthesis, or you can say ketogenesis takes place. This acetyl-CoA, increased level of the acetyl-CoA will form what? Fatty acids, cholesterol, right? So patient will be suffering from what? Hyper fatty acid synthesis. After fatty acids, it is being esterified with the cholesterol, right? So that gives you triacylglycerol, or you can say hypertriglyceridemia, or hyper excess level of cholesterol. So cholesterolemia. Does that make sense, right? So patient will be having ketone bodies, patient will be showing hypertriglyceridemia, and then you will be having hypercholesterolemia also, right? So all these things will be happening. After some time, what will happen? After some time, this is going to feedback regulate. There is too much of the acetyl-CoA that is present here, right? That is going to negatively, or you can say inhibit this pyruvate dehydrogenase enzyme. And then the pyruvate levels will start on accumulating. Now it will be rerouted towards the formation of the lactate. And then what you will see? Lactic acidosis. Keyword in the question paper, right? Lactic acidosis. What you will see? The lactic acidosis. Now, when the lactic acids are being removed from the proximal convoluted tubulets, same is the route for the excretion of the uric acid. So if you have increased lactic acid, right, that hampers excretion of what? Uric acid. This was one of the question. Somebody asked me, one of the student asked me, right, why there is increased uric acid level. There is just one, you can just imagine there is one only exit gate, right? Either lactic acid can be excreted or the uric acid can be excreted. In this particular scenario, lactic acid gets the first priority. So uric acid is being accumulated, giving to increased levels of the uric acid. Increased level of the uric acid, right? So in the von Gurk's disease, you will see so many things. Hypoglycemia, no response to the hormones, right? There are glycogens that are being accumulated. Hypertriglyceridemia, hypercholesterolemia, ketone bodies, lactic acidosis, and there is increased uric acid level. They may give you a picture of something like this, right? This belly, right? Short stature, liver and kidney enlargement, hepatosplenomegaly, fasting hypoglycemia, hyperlipidemia, hyperuricemia, right? And osteoporosis also, right? All these things you will see. Else they will give you something like this. What is the condition of this baby? Doll-like faces. This is another key point. Doll-like faces, protuberant abdomen, fasting hypoglycemia with the lactic acidosis, right? So these are the questions related to your glucose 6-phosphatase deficiency, which is causing the von Gurk's disorder, and there are so many clinical presentations. They may trick you either of the way, but you are going to sure one question sure from this, right? I hope this is going to help you, and whatever I have told you, all these topics, you are definitely going to see at least one questions from all these 5-6 topics which we have discussed. I wish you a very good luck, and remember, whether exam happens or not, right? But this knowledge is yours. If you cannot use it now, think you are going to use it in the future. You may score higher in the future. So always and always be positive. That's what I always keep saying. I'm not saying I have never been through hard times. There were tough times for me also, right? There were tough times for me also, and I have been through that. Everybody goes through that, right? But remember this, you weren't born with the ability to change someone's life, right? So don't waste it. make the best use of it, and if you have to reach to the top, right, if you can remember the spider story, it falls over and over and over. Life is not smooth. So just be hopeful, right, and just your job is to just study. You sooner or later you will get beautiful results. Trust me, that is my promise. Good luck.